In this video, we will discuss about how this sting agonist induce apoptosis in the cancer cells. So, from the heading, it is clear that it induces apoptosis through IRF3 signaling pathway. But how they come up to this conclusion, we will discuss those results in the figure that is presented on the left hand side. So first what they did is they use three kind of cell lines, that is control cell lines, TP53 cell lines and sting knockout cell lines, and then they add this reagent that induce sting in the cells, that is ADU-S100. And what they have find out is that in the case of control and TRP53 knockout, they see cell death for all the three cell lines. However, for the sting knockout, they do not see any cell death. And these things they have validated using the Western blot also, where they can see that uh, the expression of stings goes up with induction of or with the this treatment with ADU-S100 reagent. It is under clinical trial, this reagent, and thereafter they wanted to check whether it is because of from the panel A and B, they confirmed that sting induced cell death, but they wanted to check how this sting induced cell death. So what they did is they knock out the target genes that trigger apoptosis, and also they knock out some of this BH3 upregulated genes. And what they have find out is that when these BH3 upregulated proteins are knocked out, or this apoptosis upregulation proteins are knocked out, then they do not see any decrease in the cell death. But whereas in the control case, they see a decrease in the cell life populations, not cell death, cell life populations. Yeah. Whereas for these bucks and back knockout and Noxa, Puma, BIM knockout, they do not see any changes in the uh, percentage of cell lives. This indicates that the sting-induced apoptosis through sting cause cell death through apoptosis. And what they did is they measure the different BH3 target genes along with the sting target genes. And what they find out is that BH3 target genes are increased in the control case and TRP 53 knockout, and similarly they see an increase in the sting target genes such as IRF3 and this ILL6 and other genes that cause that increase when the sting pathways are triggered. So this further substantiates their point that sting will cause upregulations of BH3 proteins, and that will induce apoptosis. So this is so far they have understand that sting agonist will upregulate sting pathways, and it will trigger BH3 only proteins, and it will suppress pro-survival proteins, and the pro-apoptotic proteins goes down, and the pro-apoptotic proteins uh, will go up because the pro-survival proteins goes down, and it will trigger apoptosis. Now, the apoptosis are also triggered by two different pathways. First one is through IRF3 pathway, and second one is this NF kappa beta pathway. So they wanted to check which of these pathways is actually following or actually happening, or both the pathways are happening. So what they did is they knocked down this TBK1 and they knocked down this IRF3, and in another case they knock down this NF kappa beta, or this is the region that they use, that is GSK 8612 will down regulate this TF kappa beta 1, and if this TBK1 is down regulated, then this NF kappa beta pathways is still active. So in this case, in the panel F, what they have seen is that they seen a significant decrease in the cell life populations when they dysregulate this IRF3 pathways. Uh, Sorry, I want to correct myself. So when they dysregulate this IRF3 pathways, that is using GSK 8612, and they induce these ADU-S100, they do not see a significant decrease in the populations. Indicates that this IRF3 might be playing a role and not NF kappa beta. So in order to validate that thing, they also knock down this NF-kappa beta using TPCA1 pathways. Here, what they see it when they use the TPCA and this sting inducing reagent, that is ADU-S100, they see a decrease in the proportion of live cell populations for all the 3 cell lines. So this together, both of these results together indicates that is the IRF3 pathways are mainly triggered for the apoptosis when the sting is induced in the cancer cells. And the graph has been shown in this panel G, where we can see that when they induce when they use, and after validating the results, they check in the knockdown cells, that is they knock down IRF3, and in other case they knock down they both of them are different IRF3 knockdown cells. And what they have find out, in the case of control, the percentage of live cells goes down, but in the case of IRF3 is been deficient or it is knocked down, they see a very less cell death. So all this indicates that it's the IRF3 pathways that plays a role towards apoptosis when the sting is induced in the cancer cells, not the NF-kappa beta pathways. But researchers are expecting NF-kappa beta pathway, but they do not see any significant decrease in the percentage of live cells when this IRF3s are being inhibited, or when these NF-kappa are